In order to understand the biology, epidemiology, and management of economically important diseases affecting fruit and vegetable crops, it is important to be familiar with the basic characteristics of the major groups of plant pathogens. We'll start with a review of each major group, and this should be a review as this was covered in introductory plant pathology. There are also reference materials listed in Carmen if you find that you need them. The vast majority of microorganisms, whether subcellular, such as viruses and viroids, single cells like bacteria, or multicellular, like all the rest listed here, are not pathogens on plants. Plant microbes are highly specialized to parasitize plants and represent a wide range of diversity even within the same general group. We will discuss these in order of size and complexity, beginning with viruses and viroids and ending with nematodes. There are also a few parasitic plants, such as striga and dodder, but they are not economically important in fruit or vegetable production in the U.S. and will not be covered here. So a virus is a capsid-encoding organism composed of nucleic acids and proteins that self-assemble into a nucleocapsid. The capsid is the shell of the virus's nucleic acid component and is composed of protein. Plant pathogenic viruses come in numerous nucleocapsid shapes and sizes and with different forms of nucleic acids. The main capsid forms are rigid rods, such as with tobacco mosaic virus or TMV, which is shown in the top image on the left. Flexus rods, such as potato virus X or PVX, or icosahedrons, also referred to as spheres, as you can see for cucumber mosaic virus or CMV, which is shown in the top right image. Nucleocapsids of a few viruses, such as tomato spotted wilt virus or TSWV, are covered with a membranous envelope. These characteristics, among others, are used to organize viruses into taxonomic units. These are the major characteristics of plant pathogenic viruses. Viruses are the smallest of all known organisms apart from viroids, ranging in size from about 30 nanometers for spherical viruses to 2,000 nanometers or 2 micrometers for flexus rods. In contrast, a leaf mesophyll cell is about 50 micrometers in size. As shown in the previous slide, virus capsids may be rigid rods, longer flexus rods, or spheres. A few are surrounded with a membranous envelope. All viruses are obligate biotrophs in that they can only replicate in living cells. We talked about the virus nucleocapsid, which is composed of nucleic acids surrounded by a protein coat. Multiple types of nucleic acids are possible, and the type of nucleic acid is a major factor that defines the taxonomic grouping of viruses. Most plant viruses contain single-stranded positive sense RNA, meaning that the RNA has the same polarity as messenger RNA. Other viruses contain negative sense single-stranded RNA, while others contain double-stranded RNA, single-stranded DNA, or double-stranded DNA. Plant viruses also differ in their mode of transmission. Some viruses are spread from plant to plant purely by mechanical means. A classic example is tobacco mosaic virus, which survives a long time outside the living host and is spread by contact of the virons associated with the plant tissue or sap on hands, tools, equipment, etc. One of the rules for prevention of TMV is that workers must not smoke because TMV survives in tobacco products and can be transmitted to solanaceous plants. This is particularly important during production of tomato, pepper, or eggplant seedlings in greenhouses prior to transplanting. Since seedlings are highly susceptible susceptible to viruses, and seedlings are handled relatively often during this stage. The other major means of transmission of plant viruses is through vectors, usually insects, and the mode of transmission varies with the virus and the insect species. Transmission may be persistent through the life of the insect after acquiring the virus, or non-persistent, in which case the virus essentially contaminates the mouthparts of the insect and doesn't replicate or circulate in the insect's body. There are many variations of virus-vector interactions, and the economically important ones in fruit and vegetable crops will be described in later lectures. Plant viruses can also be transmitted through grafting a non-infected rootstock or scion onto an infected one, or through vegetative propagation. This is particularly important for crops like potatoes and garlic, and some small fruit crops like brambles, for which the planted unit is a vegetative structure of the plant and likely to build up virus titer. Even though viruses are not living organisms per se, the system adopted to classify and name them is similar to that used for other groups of pathogens. Viruses are sorted into families according to their genome type and other structural, biochemical, and/or pathological characteristics. For example, single-stranded DNA viruses with 2 icosahedral capsid particles are in the family Gemniviridae. The Podiviridae contains viruses with flexus rods and positive sense single-stranded RNA. The Pospoviridae contains viroids with circular single-stranded RNA, but no capsid proteins. Each virus family contains multiple genera, which are often named after the type member of the genus. Though the Tobamovirus genus is named for tobacco mosaic virus, but this genus contains other related viruses such as tomato mosaic virus, tomato brown rugose fruit virus, and cucumber green mottle mosaic virus. Note that the virus abbreviation is usually used after the virus name has been spelled out, especially for long-standing common viruses such as TMV or PVY. When the full name of the virus is written out, only the first word is capitalized and all are in italics. You can find more information and look up specific viruses on the website shown below. This website may not be updated recently, but the information included for most viruses is still helpful. You'll notice that many virus names include a plant name and one or more symptoms, such as tobacco mosaic virus and tomato spotted wilt virus. This has been standard naming practice that continues to this day. Viruses can cause a wide range of symptoms, as you see here. A common symptom of virus infection is mosaic, in which leaves take on a mottled appearance due to the presence of dark green and light green tissue, as in the top picture of pepper leaves with cucumber mosaic virus. The banana pepper on the right was also infected with cucumber mosaic virus, but the pepper fruit is exhibiting a necrosis symptom. Other symptoms include leaf curling and deformation, such as strapping, in which leaves are much more narrow than in a normal plant. Virus-infected plants are often stunted or dwarfed, as you can see in the lower left photo. This tomato plant also exhibited leaf distortion. Other symptoms included, but are not limited to, leaf chlorosis, color breaking in flowers, and changes in the size, shape, or form of fruits. Viroids tend to cause symptoms similar to those caused by viruses, as you can see in this picture of a viroid-infected tomato. However, viroids differ from viruses in that they are essentially naked nucleic acid without a capsid. They are mechanically transmitted as described for viruses and can also be transmitted by aphids. We now move up a significant degree of complexity from viruses and viroids to single-celled microorganisms, plant pathogenic bacteria. There are relatively few bacterial phytopathogens that cause significant economic damage in fruit and vegetable crops, but those that do can destroy a crop, either outright by killing plants, by reducing plant productivity, or by damaging fruit, leaves, or other saleable plant parts, making them unmarketable. Plant pathogenic bacteria can cause localized lesions or become systemic throughout the plant. Systemic bacteria mainly move systemically through the xylem, with the exception of phytoplasma, which are in the phloem. Many are seed-borne, although usually only those that are systemic infect the seed, that is, are present under the seed coat. The others are primarily associated with the outside of the seed coat. Plant pathogenic bacteria can build up to high numbers in lesions or the plant vascular system, reaching 10 to the exponent 10 colony-forming units per gram of tissue. Pictured here is bacterial streaming from a leaf lesion in water. These are the basic characteristics of the bacterial phytopathogens affecting fruit and vegetable crops. Most of these, and the aster yellow's phytoplasma, will be described in more detail in lectures to come. This table refers to bacterial pathogens common in the northern parts of the U.S., with the exception of Ralstonia, which is a problem in the southeastern U.S. and tropical and subtropical environments worldwide. Most bacterial pathogens of plants are gram-negative. The exception is Clavibacter, a gram-positive pathogen of tomato, pepper, potato, and a few other crops such as maize and alfalfa. Clavibacter is divided into various species or subspecies, and each is fairly host-specific. This is true of Xanthomonas, the Pseudomonas syringae pathovars, and two Erwinia species of interest to this course, Erwinia ambliovora and Erwinia trichophalia. Pectobacterium and Agrobacterium have wide host ranges. Some symptoms or signs are characteristic for these pathogen genera, although symptoms alone may not be sufficient for a diagnosis. For example, the shepherd's crook caused by Erwinia ambliovora on apple trees is diagnostic, whereas leaf spots caused by Pseudomonas or Xanthomonas may not be. The bacterial genera shown in the table cause systemic and/or local infections, local defined here as lesions, cankers, galls, etc., that remain localized and the bacteria do not enter the plant's vascular system. Xanthomonas contains species or pathovars that have systemic activity, while others do not. Most bacterial plant pathogens are favored by moderate to warm temperatures. The exceptions are pathovars of Pseudomonas syringae, which tend to cause damage at cooler temperatures and thus tend to cause most damage in the spring and fall. Bacterial phytopathogen transmission varies by genus, but nearly all are transmitted by splashing water from rain or overhead irrigation. About half of the genera are also transmitted by seed. Erwinia trichophalia is transmitted by cucumber beetles, which also serve to overwinter the bacteria. Most of the bacteria survive more than 1 to 2 years in plant debris, and some also survive in water and soil. Most Xanthomonas species are relatively short-lived in plant tissue, soil, and water, usually less than a year. This PowerPoint slide is important, and I recommend that you refer back to it as you move through the lectures for both fruit and vegetable diseases.